If you're confused about why mRNA is transcribed using the non-coding DNA strand, or it just doesn't make sense that the antisense strand is used to make mRNA, then keep watching. I'll make it make sense. It helps to keep in mind that the product of transcription and translation is protein. Proteins are very specific sequences and amounts of amino acids and are synthesized 5' to 3'. Since the 5' end of mRNA contains the untranslated region, or UTR, which contains ribosome recognition and translation initiation sequences. The 5' end also contains the start codon, which codes for the first amino acid in proteins. And the 3' end of mRNA contains the stop codon, which signals the end of translation and helps dissociate the ribosome from the mRNA, releasing the completed primary protein. It also helps to remember that DNA is double-stranded, and the two strands are anti-parallel when hydrogen bonded via the nitrogenous bases, which means that one strand is in the 5' to 3' direction, and the other is in the 3' to 5' direction. And finally, it helps to remember that DNA contains genes that code for proteins. The gene codes are transcribed into mRNA, which is the messenger of the codes, carrying it outside the nucleus to a ribosome, which will then translate the code into a protein. The 5' to 3' DNA strand is referred to as the sense strand because it contains a sequence that codes for the specific order of amino acids that will come together to make the protein, and it's in the same direction, 5' to 3', as the protein must be synthesized. For this reason, it's also known as the coding strand, and it can also be referred to as the leading strand. The 3' to 5' strand is referred to as the antisense strand because it's in the opposite direction as the mRNA and protein, so the code for the protein can't be read in the same direction as the protein has to be, because this strand is in reverse of the actual code. For this reason, it's also known as the non-coding strand, and it can also be referred to as the lagging strand. So it would make sense that the sense strand is the template strand for transcribing mRNA to make a protein, but it's not. Actually, the antisense strand is the template for transcription and therefore translation. That's because mRNA must be transcribed as a single strand 5' to 3', because ribosomes only read mRNA from 5' to 3'. So, in order to make a 5' to 3' mRNA, the antiparallel strand must be used, which is the antisense or non-coding strand. You can liken transcription of mRNA to replication of DNA, resulting in the antiparallel double-stranded DNA itself. The antisense strand is what creates a sense mRNA that will have the coding DNA from the sense DNA strand to assemble the specific order of amino acids for a protein. At first, it didn't make sense, but now see that the resultant 5' to 3' mRNA is in the same direction as the 5' to 3' sense DNA, meaning it's the exact copy of the gene from the coding strand, the only difference being uracils replacing thymines. So then, the exact copy of the gene is sent out of the nucleus to a ribosome for translation into a protein. The ribosome reads the mRNA from 5' to 3', synthesizing a protein that is 5' to 3', which is the same direction as the genetic code on the coding DNA strand. So hopefully it all makes sense now as to why the antisense DNA strand is used to transcribe a sense mRNA that will be used to translate into a protein that was coded from the sense DNA. Simple as that.